L-carnitine. It was first extracted from meat in 1905 by two Russian chemists, and for decades it sat in the background of nutritional science without much fanfare. That changed when researchers began looking at how the body actually moves fat. Fat stored in your cells cannot be burned for energy on its own. It has to be physically carried into the mitochondria, the part of the cell where energy is produced. L-carnitine is the molecule that does the carrying. Without enough of it, fatty acids queue up outside the mitochondria and go nowhere. The body naturally produces some L-carnitine in the liver and kidneys, but the majority comes from animal foods, particularly red meat, which is where the name comes from. The Latin word for meat is carnis. People who follow plant-based diets often have noticeably lower levels. When L-carnitine is present in sufficient amounts, the transport system runs efficiently and fat actually reaches the furnace. Green tea extract. It comes from the same plant as regular tea, Camellia sinensis, but what makes the extract different is the concentration of a compound called epigallocatechin galat, more commonly written as EGCG. Japanese researchers began studying EGCG seriously in the 1990s after observing unusually low rates of obesity and metabolic disease in populations with high green tea consumption. What EGCG does is inhibit an enzyme called COMT, catechol O methyltransferase, that would otherwise break down norepinephrine, the signaling molecule that tells fat cells to release their stored fat. By blocking that breakdown, EGCG keeps the signal active longer. The fat-releasing process continues beyond what would normally happen. This is one of the reasons green tea extract is one of the most studied weight management ingredients in clinical literature. A 2005 meta-analysis published in the American Journal of Clinical Nutrition confirmed significant improvements in fat oxidation in subjects who took standardized green tea extract over 12 weeks. Caffeine. It works in a completely different way from EGCG, though the two are often found together for a specific reason. Caffeine blocks adenosine receptors in the brain, which reduces the feeling of fatigue and increases the release of adrenaline. That adrenaline signal then travels to fat cells and activates an enzyme called hormone-sensitive lipase, which physically breaks apart stored triglycerides and releases free fatty acids into the bloodstream. Those acids are now available to be burned. On its own, caffeine's effect on fat burning is real but temporary. The body adapts quickly with regular use. Paired with EGCG, however, the combination performs better than either compound alone. The EGCG keeps the released fat from being reabsorbed while caffeine keeps triggering the release. The pairing was documented in a landmark 1999 study at the University of Geneva, which found the combination increased 24-hour energy expenditure significantly more than either compound separately. Capsaicin. It is the compound that makes chili peppers hot. The burn you feel on your tongue when you eat a raw chili is capsaicin binding to pain receptors, specifically the TRPV1 receptor, which exists not just in your mouth but throughout your body. When capsaicin activates these receptors, it triggers the release of catecholamines, the same family of signaling molecules as adrenaline. The result is a temporary increase in body temperature and an acceleration of fat oxidation. Researchers in the Netherlands published findings in 2003 showing that capsaicin consumption before meals significantly increased fat burning in subjects compared to a placebo group. What makes capsaicin distinct from stimulant-based compounds like caffeine is that it generates heat through a different mechanism, a process called thermogenesis, without raising heart rate to the same degree. Cultures in Mexico, India, and large parts of Southeast Asia have consumed chili-based foods daily for centuries, long before the science existed to explain what was happening metabolically. Conjugated linoleic acid. It is commonly shortened to CLA, and it is a naturally occurring fatty acid found primarily in the meat and dairy of grass-fed ruminants. cows, sheep, goats. The concentration of CLA in meat from grass-fed animals is substantially higher than in grain-fed ones, sometimes by a factor of 3 to 5. What CLA does in the body is target fat storage at the cellular level. It activates a protein called PPAR-alpha, which shifts the body's preference toward burning fat rather than storing it. More specifically, it appears to reduce the activity of an enzyme called lipoprotein lipase that normally helps fat cells pull fatty acids from the blood and store them. A Norwegian study conducted in 2000, often cited as a turning point in CLA research, found that subjects taking supplemental CLA over 12 weeks lost significantly more body fat than the placebo group without changes to diet or exercise. Unlike most fat-burning compounds, CLA's primary effect is on fat storage rather than fat release, which is a meaningful distinction from what L-carnitine or caffeine do. Berberine. It is a bitter yellow compound extracted from several plants, including barberry, golden seal, and Oregon grape, and it has been used in traditional Chinese and Ayurvedic medicine for over 3,000 years. Modern research has focused on its metabolic effects, particularly its ability to activate an enzyme called AMPK, adenosine monophosphate activated protein kinase, which functions as a master metabolic switch in the body. When AMPK is activated, the body shifts into a fuel-burning state. It increases the rate at which cells absorb glucose, reduces fat production in the liver, and improves insulin sensitivity. A 2012 study published in the journal Metabolism found that berberine outperformed a placebo in reducing body fat and improving metabolic markers in obese subjects over 12 weeks. What is unusual about berberine is the breadth of its metabolic action. It touches blood sugar regulation, fat oxidation, and cholesterol metabolism simultaneously. It connects back to some of the mechanisms seen with green tea extract but operates through an entirely different enzyme pathway. Forskolin. It comes from a plant called Coleus forskolii, native to India and parts of Southeast Asia, and it has been used in Ayurvedic medicine for centuries, primarily for heart and lung conditions. The compound was largely unknown to Western science until the 1970s, when researchers at the American Heart Association began studying its effects on cellular signaling. Forskolin works by directly activating an enzyme called adenylyl cyclase, which increases levels of cyclic AMP inside cells. Cyclic AMP is a cellular messenger that triggers hormone-sensitive lipase, the same enzyme caffeine activates through a different route. The fat breakdown pathway gets switched on. A 2005 randomized controlled trial from the University of Kansas, one of the first high-quality studies on forskolin in humans, found that male subjects taking standardized Coleus forskolii extract over 12 weeks experienced significant reductions in body fat percentage and increases in lean mass. The study also noted increased testosterone levels, which may contribute to the body composition shift independently. Synephrine. It is extracted primarily from the bitter orange fruit, Citrus aurantium, a plant that has been used in traditional Chinese medicine since at least 100 AD, where it was called zhi shi. Synephrine became widely discussed in the early 2000s as a structural cousin to ephedrine, a powerful stimulant that was banned by the FDA in 2004. The two compounds are similar in mechanism, but different in how aggressively they act on the cardiovascular system. Synephrine binds to beta-3 adrenergic receptors, which are found primarily in fat tissue. Activating these receptors increases fat breakdown and thermogenesis without the same level of heart rate elevation associated with ephedrine. Research published in the International Journal of Medical Sciences in 2012 reviewed multiple clinical trials and found consistent evidence for synephrine increasing metabolic rate and fat oxidation. It is often combined with caffeine and EGCG in commercial formulations, where each compound reinforces a different part of the fat-burning process. Release, transport, and oxidation, each covered by a different ingredient.